So greetings everyone and welcome to the Science of Childhood Cancer Lecture Series hosted by the St. Jude Comprehensive Cancer Center. My name is Seth Carroll. I'm a clinician researcher in the Leukemia Lymphoma Division here at St. Jude. And it's my great pleasure today to welcome our speaker, Jun Yang, who I've known since my time as a fellow here. Before I formally introduce him, I'd like to note that you can submit your questions at any time during the talk using the chat function and sending questions to all panelists. I'd also like to let you know that if there are additional questions, Dr. Yang has kindly offered to stay on for some time after the top of the hour if anyone would like to stay for further discussion. And now I'm delighted to introduce Dr. Yang. Jun received his bachelor's in science and master's in molecular human genetics from Shanghai Jintong University. He then received a PhD in chemistry from Purdue before coming to St. Jude for his postdoc. There, he worked in the lab of Dr. Mary Relling and was incredibly productive with many seminal discoveries in the areas of second malignancies and genetic variants which predispose to human acute lymphoblastic leukemia. He is probably most well known for his work in pharmacogenomics, where he has made many important discoveries in how inherited and acquired variants impact chemotherapy toxicity and efficacy. He joined the faculty here at St. Jude in 2010 and rose quickly through the ranks, was promoted to the rank of full member in 2019, and has served as vice chair of the Department of Pharmacy and Pharmaceutical Sciences since 2021. Jun's research program has been highly productive with over 120 published original manuscripts and numerous NIH and foundation grants. And Jun is also a highly sought after mentor. On a personal note, I will say that Jun is a fantastic collaborator and always offers insightful advice about both projects and manuscripts. Today, Jun will focus his talk on his work in pharmacogenomics for ALL. I know I speak for all the attendees when I say that I'm very much looking forward to the talk. And please join me in welcoming Dr. Jun Yang.
Well, thanks for the generous introduction. I also want to thank Charlie and Charles and the Cancer Center for having me on this fantastic seminar series. So the disease I'm interested in is acute lymphoblastic leukemia. I probably don't have to go through extensive introduction to this audience, but I just want to say that this is still the most common cancer in children, about 3,500 new cases every year. And it's characterized by this uncontrolled growth of the undifferentiated immune cells, which you can tell under the microscope of the bone marrow aspirates compared to the normal bone marrow microenvironment. So when I first came to St. Jude 2006, I was taught there were about a handful of subtypes within pediatric ALL. First off, you split them into TAL versus BALL based on immunophenotype. And then there are a number of sentinel genomic abnormalities that define other subtypes, such as ETV6 RUNX1, arising from the 1221 translocation, TCF7 PBX1, et cetera, et cetera. So 14 years later, through a series of collaborative projects between St. Jude and the Children's Oncology Group led by Steve Hunger, Mignon Lo, particularly Charles Mugan here at St. Jude, we have a much better understanding of the genomic complexity of acute lymphoblastic leukemia in children and in adults. So in BLL alone, there are 23 molecular subtypes, as shown on the T-SNP plot on the left, and also the classification flowchart on the right. I think there are about another dozen or so subtypes within TLL as well. But the diversity of ALL not only just exists in the leukemia biology, there is also incredible intrapatient variation in drug-related phenotypes. For example, there is intrapatient variation in the response to leukemia drugs, the leukemia intrinsic drug toxicity, drug sensitivity, as well as acquired resistance and relapse. These phenotypes are oftentimes driven by somatic genomic abnormalities. There are also germline genetic polymorphisms that can influence a patient's ability to metabolize the drug, pharmacokinetic profile of specific medication, 04 acute toxicity during therapy, as well as late effects in ALL survivors. So pharmacogenetics is aimed to evaluate how an individual's genetic makeup corresponds to the response to a specific drug. And the ultimate goal of pharmacogenetics is to individualize medical treatment to each patient, maximizing the effectiveness while minimizing side effects. So pharmacogenomics ALL obviously has a lot to do with germline genetic variants and its impact on ALL treatment toxicity, as very well exemplified in the context of thiopurine metabolism and toxicity in children with ALL. And myself and others have talked about that quite a bit in the past, so that's not going to be the focus of my talk today. Instead, I want to spend the time and discuss some examples from my own work here at St. Jude, looking at the relationship between somatic genomic abnormality and how they impact leukemia drug sensitivity. And I have 3 examples I'm going to share with you guys for the next 40 or so minutes. So to do pharmacogenomics, the first step is to really define the drug response phenotypes. And to do this, a few years ago, led by Unraveling and Minyoung Lo, we put together a P50 grant application to the NIGMS to perform large-scale ALL pharmacogenomics. And this is a specific project led by myself and Dr. Bill Evans, is aimed to include about 1,000 ALL cases, 500 children, 500 adults. We want to define drug sensitivity phenotype of these 1,000 cases, tested about somewhere between 15 to 20 drugs. And in parallel, we also want to perform comprehensive genomic profiling of each one of the 1,000 cases, RNA-seq, whole genome sequencing, exome sequencing, SNP array. End of the day, we want to identify in a comprehensive fashion the molecular determinants of leukemia drug sensitivity and construct what we call a gene-drug connectivity map. We envision that this connectivity map will be able to help us to develop the next generation of ALL therapy here at St. Jude and elsewhere. And obviously, this is a large collaboration involving not only investigators here at St. Jude, but also Children's Oncology Group and a number of colleagues at MD Anderson, ECOG, and University of Chicago for the adult ALL populations. So ALL pharmacotyping or drug sensitivity profiling is not new to St. Jude. Bill actually started doing this in the late 90s and early 2000s. He primarily focused on cytotoxic drugs, prednisolone, vincristine, asparagine, and thiopurine, and coupling the drug sensitivity profiling with molecular profiling techniques such as gene expression profile. His group actually has uncovered fascinating biology linking drug sensitivity to genetic abnormality, as illustrated by a series of seminal publications listed down below at the bottom of the slide. So our pharmacotyping platform also evolved over time, and this is the current assay that we use to test drug sensitivity on primary ALL cells. It is a co-culture system where we use mesenchymal stem cell as a feeder layer. This actually allows much better survival of leukemia cells ex vivo. It is a high-content single-cell imaging platform that we currently use to determine cell viability, morphological changes, and in response to drug exposure, with the flexibility to add a number of other intracellular markers. Because this is the fluorescent-based platform, there are 11 colors that we can play with. And we currently actually achieve a fairly good throughput with the 384 well formats. We are able to test about 44 drugs per patient using about 20 million cells. And this is a 96-hour assay. And currently, this is operated by 2 very talented researchers in the pharmacotyping resource, Brandon Smart as well as Noemi Reyes. So between Bill's lab, assisted by Christine Cruz, and also my lab, we have now collected ALL pharmacotype or drug sensitivity data for over 1,300 cases. As illustrated here, divided into newly diagnosed cases, over 1,200 of them, and also relapsed refractory cases, 158. If you look at the newly diagnosed cases, a majority of them are pediatric, about 900, and we have 321 from adult ALL. In the relapsed refractory cohorts, it's a little bit more even split between pediatric and adult population. So this data sets will actually give us a unique opportunity to understand the biology of age-related differences in drug response in ALL. So we have 2 classes of agents we have tested. the cytotoxic drugs, including asparaginase, vincristine, glucocorticoids, and a number of antimetabolites. Since 2015, we also added a large number of targeted therapeutics, including tyrosine kinase inhibitors, targeting ABL, BTK, MEK, JAK2, etc., and also BH3 mimetics, specifically going after BCL2, BCL-XL, HDAC inhibitors, proteasome inhibitors, and an antibody conjugate drug, inotuzumab. So I don't have time to go through all the drugs and for all the ALL cases we have tested, but I want to show you this slide to really illustrate the degree of interpatient variability in drug sensitivity of leukemia cells in pediatric and also adult ALL. So each plot here is a drug looking at the histogram showing the variability of the drug sensitivity profile acquired from each individual patient. And there are a total of 16 showing here. As you can see, across all drugs, there's always a wide range of variation in terms of the leukemia sensitivity to that agent. And I want to draw your attention to 3 different drugs representing 3 characteristic patterns of interpatient variability in this drug response phenotype. The first one is daunorubicin. The LC50 distribution really looks like a normal distribution, right? You have a majority of the cases with intermediate response, and then you have some of them with extreme resistance, a small number of them with extreme sensitivity. And the second pattern we see is represented by asparaginase. So now you have a very large proportion of patients with extreme sensitivity to this drug, and primarily children, very small percentage of adults. You also have a large proportion of the patients that are very resistant to this drug. And then you have a relatively smaller portion of the individual showing intermediate response to asparaginase. And the third drug representing the third pattern of variation is the dasatinib. So now you have a majority of the patients completely resistant to this drug. You have some of them are very, very sensitive, but only the remainder of the patients, only a small proportion with intermediate response. So for things like the dasatinib, it's almost on-off switch. You're either very sensitive, you're not, or you're not sensitive at all. So these 3 patterns really are representative across the 16 drugs that we have tested so far. And this is really a source of discovery for us, as I will illustrate for the remainder of my presentation, as how we go about discovering pharmacogenetic variants driving these variations. So focusing on the pediatric cases, we decided to do a large analysis to comprehensively profile drug sensitivity across modern molecular taxonomy of ALL. We want to associate ALL drug sensitivity in vitro with the MRD, which is response in vivo to ALL therapy, and also look at the impact on treatment outcomes. To do this, we collaborated with our clinical investigators here at St. Jude, put together a cohort of 805 children from total of 15, total 16, and total 17. The phenotype, as I mentioned, is drug sensitivity for 18 different agents. And the genotype here, or the genomic data here, is RNA-seq-based LL subtype classification in collaboration with Charles Milligan, Catherine Roberts, and many others. And this analysis is led by a very talented clinical fellow, Shang Li, in my lab, in collaboration with Wenjing Yang, a bioinformatic research scientist in our departments. So first off, let's look at the LL subtype in the pharmacotyping cohorts. Showing on the left is a testnet plot of the pan-ALL cohort that Charles published in 2019, describing molecular subtypes of 1,988 ALL patients, primarily pediatric with some adults as well. Each dot is a patient. As you can see, based on gene expression pattern, you can identify a range of molecular subtypes, oftentimes characterized by their translocation, ETV6 from, for example, DUX4 rearrangement, etc. Now, if you look at the ALL pharmacotyping cohort, 805 patients, you can immediately appreciate we also observe similar pattern of LL subtypes compared to the pan-LL cohort, although the exact location of the dot is different because it's a different data sets, but the clustering pattern is very, very consistent. ETV6 RUNX1, ZNF3-4 rearrangement, DUX4, et cetera, et cetera. So this is the LL subtypes we are able to identify in this cohort of 805 patients. ETV6 RUNX1 and hyperdiploid remain the most common, which is not surprising. But then you also have many other more rare subtypes, especially some of the more recently discovered ones, DUX4 rearrangements, CRF2, either or not would have the pH-like gene expression signature, and also early T cell precursor. So this is a summary of the drug sensitivity profile across 23 different LL molecular subtypes. Going from top to bottom, each row here is a drug, vincristine, penobenostat, prednisolone, so on and so forth. Going from left to right, each vertical column here is a molecular subtype, ETV6 RUNX1, ETV6 RUNX1-like, hyperdiploids, so on and so forth. For the sake of time, I'm not going to go through each drug subtype combination. What I will do is to show a couple of examples, really just to give you a taste of what this data set enable us to, type of questions we're able to ask and answer. So the first example is 3 low-risk subtypes, DUX4, ETV6, RUNX1, and also hyperdiploid. These 3 subtypes are very well known to have some of the best prognosis in pediatric ALL, with event-free survival and overall survival approaching 95, if not higher, at 5 years. So you would assume that they have a very similar pattern of sensitivity to the cytotoxic drug we give. But what you can see here is DUX4, which is highlighted in pink. This subtype is very, very resistant to asparaginase compared to ETV6 RUNX1 or hyperdiploid. Matter of fact, for ETV6 RUNX1, vast majority of patients with that subtype are very, very sensitive to this drug. In hyperdiploid, you actually have a significant proportion of patients with some level of resistance to asparaginase, but overall, it's still much more sensitive compared to patients with DUX4 rearrangement. What about prednisolone? What's really interesting is with ETV6 RUNX1, like asparaginase, almost completely sensitive to prednisolone. And with hyperdiploid, now you have 2 clusters of individuals. Some of them are very sensitive, but then some of them are significantly resistant. And this is similar to DUX4 with the split between sensitivity versus resistance in terms of prednisolone. However, DUX4 patients are exceptionally sensitive to vincristine. The vast majority of the patients with that subtype have a very low LC50 to vincristine compared to ETV6, RUNX1, or hyperdiploid. What about the targeted drugs? What we're showing here is venetoclax LC50 on the y-axis. As you can see, hyperdiploid patients seem to be quite sensitive to this drug compared to DUX4 and ETV RUNX1, although this is a drug that we currently are not using in the frontline setting for pediatric ALL, but does point to potential therapeutic opportunities when we think about next generation of trials here at St. Jude's. So another example is the comparison between within TLL. So there are 2 different subtypes, those with the early T precursor phenotype versus non-ETP ALL. So Dario Campana, when he was still at St. Jude many years ago, discovered ETP as a unique entity within TLL with exceptionally poor prognosis. So what does that correlate with the drug sensitivity pattern that I'm about to describe to you? So if you compare non-ETP TLL versus ETP, ETPs are very, very resistant to prednisolone. They're also very resistant to cytarabine. They're more resistant to mercaptopurine as well. However, there is a trend for them to be more sensitive to venetoclax, although because of the small number, we didn't observe a statistically significant difference here. So this is consistent with the known fact that ETPs tend to have high minimal residual disease after induction therapy, probably because they were resistant to the drug we used during remission induction phase of ALL therapy. So what about correlation of in vitro drug sensitivity to drug response in vivo? So here we're looking at two different phenotypes, the minimal residual disease, which measures the in vivo response to LL therapy. And there are two different time points here. One on the left is day 15 MRD, which is about 2 weeks of LL therapy upfront. And then day 46 is 6 weeks of induction therapy with a combination of agents given here. And the drugs we give during induction therapy are highlighted with asterisk here on both plots. So what you can appreciate is, so each row here is a drug, and if it's highlighted in red, that means it's a statistically significant correlation between drug sensitivity and MRD. And if it's to the right of this vertical dashed line, that means the drug resistance phenotype is associated with higher minimal residual disease at these two time points. So what you can appreciate is asparaginase and glucocorticoids had the strongest influence in MRD results on day 15 and also day 46. What's also interesting is thiopurine and thiaguanine and mercaptopurine, the two drugs that we did not use during the first 15 days of therapy. They also showed very strong correlation with MRD results, indicating the sensitivity to this drug are probably related to intrinsic mechanisms to maybe propensity to apoptosis. And that's why they are associated with MRD or general response to LL therapy for the first phase of treatment. So we also performed the unsupervised clustering analysis using the LL pharmacotype datasets. We were able to identify 6 different groups. So each row, again, is a drug. Each column here is a patient. As you can see, there are 6 groups on this heat map. So cluster 1, 2, 3, 4, as listed from left to right. And the panel on the right here indicates the subtype classification for each cluster. So I just want to draw your attention to cluster 1. So this group right here is characterized by sensitivity to dasatinib, the third row from the top. So this is the only group with a substantial sensitivity to dasatinib across all 6 different groups. And if you look at the molecular subtypes within cases in cluster 1, they're mostly BCR-ABL or TLL. And cluster 3 and 4 are most drug sensitive across different, especially for the cytotoxic drugs. And they're overrepresented with hyperdeploy ALL and also ETV6 RUNX1 ALL. So the question is whether or not the drug sensitivity profile that we're able to identify using in ex vivo assay, does it have any prognostic impact? So when we look at the differences in event-free survival across different drug sensitivity-based cluster, cluster 1, which the one overrepresented with TLL and BCR-ABL, actually had the lowest event-free survival across all different groups that were identified based on sensitivity. And what's really interesting is when you look at the events within cluster 1, all of them occurred in patients with TLL, and all the BCR-ABL positive patients actually fared relatively well on total 15 and total 16. So what is going on with the dasatinib and the TLL? What we did here was to pull out all the TLL cases on total 15 and total 16. This is 97 patients. And then we look at their sensitivity to dasatinib. Clearly, there are 2 different clusters. Some of them are very sensitive, some of them are very resistant. This goes back to the drug sensitivity pattern I mentioned before for this drug. It's either on or off. And now you split the patients based on their dasatinib sensitivity into sensitive versus resistant. You see that there's a very significant difference in survival on total 15 and the total 16. Patients who are sensitive to dasatinib had a particularly poor prognosis compared to patients who were dasatinib resistant. So again, pointing to the prognostic value of this pharmacotype. In fact, dasatinib sensitivity is significantly associated with the EFS even after adjusting for MRD. So this really drives us to start thinking more about the dasatinib sensitivity in TLL, wanting to understand the biological basis of this variation in drug response. Dasatinib, for those of you who are not familiar with, was originally developed by Bristol Myers Squibb as the ABL inhibitor, showing the crystal structure of BCR-ABL. Dasatinib is the small molecule in the middle. And this drug is highly efficacious for patients with the pH positive BLL or CML. If you look at the BLL cases that we have tested, vast majority of them, if they're sensitive to dasatinib, they're positive for the BCR-ABL fusion, or they have fusion gene involving other member of the ABL class genes, such as PDGFRB, PDGFRA, etc. But in TLL, 41% of them are sensitive to dasatinib. The prevalence is about 44% in pediatrics, 16% in adults. What's really interesting is in this cohort, nobody had BCR-ABL fusion. So the sensitivity is clearly not driven by BCR-ABL fusion gene. And so this prompted us to start doing a series of experiments to understand the biological driver of the dasatinib response in TLL. And this project is led by three individuals in the lab. Yoshihiro Gocho, who is a postdoc in the lab who recently moved back to Japan, and Augie Zhang, who is a technician in the lab and now is a PhD student at Mayo Clinic, and the Jian Zhong, who actually will be graduating next Monday with his PhD from UTHSC. And part of the results are published in 2021 by us. So in collaboration with the Jiang Yu's group, particularly the postdoc Jingjing Liu in computational biology department, we applied their Bayesian-based net bit analysis. It's a system pharmacology analytical pipeline to look at the driver for the dasatinib sensitivity in this subtype of ALL. What they did was to first construct the TLL signaling interactome using gene expression data to understand the gene-gene interaction or the correlation or anti-correlation of their gene expression pattern. And from that, they were able to estimate the activity of each of the 20,000 genes in cases that are sensitive to dasatinib versus those that are resistant. And this comparison will give us the candidate list of genes that are driving resistance or sensitivity to dasatinib in this cohort. So altogether, we identified 461 genes associated with the dasatinib sensitivity with a p-value cutoff of 10 to the negative 5. The top hit is actually a tyrosine kinase called LCK with a p-value of 10 to the negative 8. And if we put the 461 genes identified in this analysis into a pathway database, the pre-TCR signaling was really the top process that's enriched in the dasatinib sensitive TLL. And I'm showing the major components of the pre-TCR signaling pathway. And the ones highlighted in red are the molecules that are significant in our NetBit analysis, differentiating the dasatinib sensitivity versus resistance. So LCK, pre-TCL alpha, a number of different subunits of CD3 and CD247 and ZEP70 as well. So we also performed the phosphophotolog experiment to validate the phosphorylation of LCK, ZEP70, and CD247. As you can see, cases that are sensitive to the dasatinib had a higher level of phosphorylated LCK, ZEP70 and CD247 at the baseline, and this level went down as when you increase the concentration of dasatinib in vitro. And if you're resistant, you never had any phosphorylation from the get-go, obviously that won't change even if you put dasatinib in tissue culture. So why is LCK important in TLL? To talk about this, we first have to review the differentiation process of a normal T cell development. It goes from the very immature precursor cells and enter this stage called double negative. They're negative for CD4, negative for CD8, then 1, 2, 3, 4 different stages, and then enters the double positive stage for both CD4 and CD8, and then they will split into the single positives, either for CD4 or for CD8. And there are a number of checkpoints that involve LCK during this process. The first one is called pre-TCR checkpoints. And this is triggered by the activation of a pre-TCR alpha gene. And once this is activated, it actually phosphorylates LCK, and this triggers a very strong proliferation signal. This really gives the normal T cells a signal to expand, because now we need a large pool of T cells for downstream selection, either positive or negative. And the second place LCK is important is at the TCL alpha beta checkpoints. So here you have the canonical TCL alpha expression. You form this complex with TCR beta, and triggers again LCK activation. But now you can send either a positive selection signal, which triggers proliferation, or a negative selection signal, which might lead to apoptosis eventually. So what's really important for TLL is the pre-TCR activation, which is almost always linked to a strong proliferation signal. So we wanted to test this in an experimental system. And when we look at the genomic abnormalities identified in our dasatinib-sensitive TLL, there are actually a couple of cases with a unique translocation. It's a fusion gene between two transcription factors, TCF7 and SPI1. And it turns out this actually provided a very interesting experimental system for us to test the hypothesis that LCK activation and dependency is related to T cell developmental arrests in leukemia. So as I said, it's a fusion of two different transcription factors, TCF7 and SPI1. So TCF7 is abundantly expressed during T cell differentiation, but SPI1, which encodes PO.1, is not expressed at all during T cell development. By fusing these two things together, now you have abnormal expression of SPI1, which again, as I mentioned already, encodes PO.1. And this completely changes the transcriptional landscape of ALL cells expressing this fusion gene. And what happened is, so now we take mouse LSK cells and we transduce them with the TCF7 SPI1. We allow them to differentiate in vitro using an OP-line co-culture system. If it's an empty vector, you can see the x-axis here is the CD44, y-axis is CD25. We use this to define the populations into double negative 1, 2, 3, 4. So if it's an empty vector, they can go through the entire process and eventually stuck in DN4, about 78%. But now if you overexpress TCF7 SPI1, you see majority of the cell actually gets stuck in the DN3 stage, about 63.4. Only about 20% of the cell were able to go through into DN4 stage. And if you measure the phospho-LCK level in the TCF3 SPI1, expressed cells is much, much higher compared to the empty vector. And this also renders exceptional sensitivity to dasatinib in the primary TLL cells carrying this specific fusion. So does this work in patients? At the time we were sending the manuscript out for review, a former postdoc of mine, Hui Zhang, who is also a pediatric oncologist, returned to China a few years ago. So he called me talking about a specific patient that he had turned out to be positive for TCF7 SPI1 fusion. The patient at that time was actually becoming refractory to conventional cytotoxic drugs. The patient actually had a very high WBC to start with, and also CNS leukemia, which is problematic for TLL. And at day 42, the patient stopped responding to cytotoxic drugs. And MRD was about 17.2% in the bone marrow by flow, which is very high. So apparently they need to do, and clearly they need to do something to the patient, otherwise it's going to be difficult to control the leukemia. So Hui actually was able to get the leukemia cells and test the sensitivity to dasatinib and imatinib shown on the top. As you can see, the patient was highly sensitive to dasatinib in the in vitro assay. And using phosphoflow, you can also see the LCK phosphorylation, the one in the middle. And if you treat with the dasatinib in vitro, you can block the LCK phosphorylation. So and that was good enough for the treating physician to decide that they need to try the dasatinib on this patient. So before that, while they were doing all the phosphor flow, they're doing the in vitro experiments, they had to give the patient something, and they gave him dexamethasone. And for 3 days, as you can see with dexamethasone, WBC just kept going up. There was no turning back, and you could not control the patient. And as soon as the dasatinib was administered to the patient, within 24 hours, you see a huge drop in WBC in the peripheral blood, along with the blast percentage in the peripheral blood. Within 4 days, leukemia blast percentage dropped from 51% to 7%. And the patient actually is still on the dasatinib now about a year and a half, with no notable toxicity. Patient is in remission. So fingers crossed, this patient will remain leukemia-free. So what we learned about the dasatinib sensitivity in TLL, 44% of them are sensitive in the pediatric population. And pre-TCR LCK activation is the reason for sensitivity in this subset of TLL. We also performed genome-wide CRISPR knockout screen in collaboration with the Kim Stagmeyer's group at the Dana-Farber to show that LCK dependency is unique to TLL compared to other cancer cell lines. And we showed that TCF/1 fusion can block differentiation, activates LCK. That's why they're sensitive. And I didn't have time to talk about that. We subsequently have done a lot of single-cell profiling experiment to show LCK activation is heterogeneous within TLL, and BCL2 activation is a potential mechanism for dasatinib resistance in this subset, pointing to possible combination therapy using LCK inhibitor and also BH3 mimetic. So for the last section of my talk, I want to switch gear and talk about some unpublished results related to the immunotherapeutic agent blinatumomab. So this is a drug that's really has garnered a lot of attention in the past couple of years because it's highly efficacious in refractory and relapsed ALL in both children and adults. So it's a bispecific antibody because it recognizes CD3 molecule on the T cells. It also recognizes CD19 molecule on the B-ALL cells. And blinatumomab can bring the T cells and B-ALL together. And as a consequence, it will release activates the T cell, release a lot of cytokines, porphyrin, granzyme, et cetera, et cetera. At the end of the day, you will see this tumor lysis, apoptosis, and eventually kill the leukemia cells. This is mechanism of action for blinatumomab. So as I mentioned, it's a highly efficacious. So Children's Oncology Group recently conducted a randomized trial, ALL 1331, to compare the efficacy of blinatumomab in first relapse patients against intensive chemotherapy. It turns out that the blinatumomab was much advantageous compared to intensive chemotherapy. And what's really interesting is if you look at the MRD status at the end of the first cycle of blinatumomab, although majority of the patients did benefit from this drug, still about 25% of patients did not respond to blinatumomab at all. And even after one more cycle of blinatumomab, these patients persisted as non-responders. So that really prompted us to think what will be the biological basis for this interpatient variation in sensitivity to this immunotherapeutic drug. So Eugene Lee, a postdoctoral fellow in my lab, decided to perform a CRISPR-Cas9 screen to identify leukemia intrinsic factors associated with the resistance to blinatumomab in TLL. So the assay we set up is this is a co-culture system. You have to have CD3 positive T cells and normal T cells. You have your leukemia cells labeled with fluorescent dye, and then you add blinatumomab at the different concentration. Incubate for 24 hours. Then you add DAPI. You do flow cytometry, focus on the PE positive population, which is leukemia, and then within this population, you look at DAPI to identify those who are dying or undergoing apoptosis. So the CRISPR-Cas9 experiments, we used the NOM6, which is DUX4 rearranged the BLL. The cells were transduced with the glyco library and is with a low MOI, such that each cell will only have one guide RNA. And now you makes the effector and target cells a 1-to-1 ratio with 2 different concentrations of blinatumomab or no blinatumomab at all, and incubate them for a certain period of time, and then you harvest the cells that survive the blinatumomab exposure, and extract the genomic DNA, and you quantify the guide RNA that's present in the surviving cells. So the idea is if knocking out a specific gene render drug resistance, the guide RNA should be enriched in the cells that actually survive the blinatumomab treatment. So this is the CRISPR-Cas9 result. Each dot is a gene. As you can see, CD19 is the top on the list. Of course, this is expected because CD19 is the target of blinatumomab. And then also CD81, which is a chaperone molecule important for CD19 surface presentation of BLL. But what's really interesting is the CD58, and I will spend the rest of the time talking about why this gene is important. So now, if you look at CD58 knockouts compared to the parental, you plot on the y-axis is BLL survival in the co-culture system in response to different concentrations of blinatumomab. And CD58 knockout clearly led to very marked resistance to this drug, almost on par with the CD19 knockouts shown on the left. And now if you reintroduce the CD58 to the knockout cell line, you can completely reverse the drug resistance phenotype, suggesting this is a CD58 specific effect that we're seeing here. So CD58 is a molecule expressed on BLL. It actually interacts with CD2 on the T cells. And this CD2, CD58 signaling axis is important for co-stimulation of T cell and for its activation, as reported by others previously, but not in this context. So now the experiment we did was to block CD2 on T cells or block CD58 on B cells using antibody specific to each one of those two molecules. As you can see, if you block either one of the two, you make the BLL cells completely resistant to blinatumomab, even in the presence of T cells. So because we're asking Drew, we're interested in genomics, we're asking the question of CD58 expression. The variation in CD58 expression across different ALL subtypes, again, 23 of them. As you can see, it is quite a range. The highest is observed in TCF3 PBX1. In the lowest is observed in patients with a specific mutation in PAX5 gene, the P80R mutation. It's a missense mutation. So the difference in CD50 expression between the highest group and lowest group is almost tenfold, which is quite substantial. So PAX5 P80R is a recurrent missense mutation in the PAX5 gene and identified by Charles Mullison's group in 2019. And it's about, I would say, less than 10%, or less than 5% in fact, of ALL. And it's almost always accompanied by a frame shift or somatic loss of the wild type allele. So these patients have a homozygous loss for PAX5 activity. And it defines a very clear gene expression pattern. So it's that's why it's classified as a specific ALL subtype. So in order to understand the effect of the PAX5 P80R on CD50 expression and also blinatumomab sensitivity, we actually use CRISPR editing to introduce PAX5 P80R mutation in the 697 cells. So now we have 3 different clones, wild type for PAX5. We have a heterozygous loss of the PAX5, introducing frame shift on one allele. We also have a clone where we have PAX5 P80R on one allele and then the frame shift on the other allele. So this really very nicely recapitulates the PAX5 loss in patients. So now we look at blinatumomab sensitivity. So the blue curve here is the wild type clone, very sensitive to blinatumomab. And now if you have a heterozygous loss of PAX5, which is orange line, you become resistant. If you're a homozygous loss of PAX5, you're also very, very resistant. Now, if we do a rescue experiment where we reintroduce CD58 to the clone with PAX5 P80R and also frame shift on the wild type allele, you can rescue this phenotype in terms of blinatumomab sensitivity to a large degree. We're also able to identify a number of primary ALL leukemia blasts of different subtypes and tested blinatumomab sensitivity as well as CD58 expression. As you can see, there is a range of CD58 expression with PAX5 being the lowest. Now you tested the blinatumomab sensitivity using the in vitro assay we developed. So we have only 4 cases. I can't do any statistical analysis. But the y-axis is the LC50 for blinatumomab, x-axis is CD58. There is a nice trend for negative correlation between the two, consistent with our hypothesis that CD58 is important for sensitivity to this drug in BLL. So why is PAX5 important in CD58 expression? So we use the 69.7 clones that we engineered to ask this question by doing a number of epigenomic profiling experiments. First off, we're looking at PAX5 binding using ChIP-seq. As you can see, if you're a wild type for PAX5, you have a very strong PAX5 binding site in the intronic region of CD58. If you lose one allele, you still have some binding, but not as prominent. If you have homozygous loss, PAX5 binding is completely gone. And this is consistent with the loss of K27 acetylation mark and also K4 methylation mark, suggesting that the removal or loss of active enhancer here in the intronic region of the CD58. And this is also confirmed with TAXIC results as well. We also did ChIP qPCR for PAX5 binding, for histone modification, again, consistently point to the notion that PAX5 is a transcription factor driving enhancer activation at this locus. So PAX5 also affects the CD19 transcription, but not to the same degree as it does to CD58, as illustrated here. SEMSEP experiments, PAX5 ChIP-seq, histone modification. As you can see here, with the loss of PAX5, you have some degree of loss, but not nearly as much as you saw with CD58. So we also did a CRISPR-I experiment to block this PAX5-driven enhancer in CD50 as a CD58 locus. And you can see that the CD58 expression is indeed compromised if you block this enhancer at this locus. Did an experiment to look at T cell activation and also proliferation with or without CD58 knockouts. So on the top panel is the CD69 staining as a marker for T cell activation. And the blue bar is the parental NOM6 with the blinatumomab, with the T cell co-incubation, very significant activation of T cell. If you knock out CD19 or orange bar, there's no activation whatsoever. If you knock out CD58, it's also largely compromised. And this is true if you focus on the CD25 as a marker for T cell activation. And we also look at the CD4 positive T cells and CD8 positive T cells with largely similar results. And this is we look at T cell proliferation using a fluorescent dye assay. You see very similar results, again pointing to the importance of CD58. So lastly, since we got the CRISPR library, we have the Cas9 cells. We thought, why don't we do a transcription factor library screening to identify all the regulators of CD19 and also CD58 using CRISPR screen. And you can see the top on the list for CD58 is actually Wang-Swan, and PAX5 ranked the number 2 on that list. If you look at CD19, it's completely separate transcription factors, important for the expression of CD19 here. So what did we learn from blinatumomab resistance or pharmacogenomics? We did the CRISPR genome-wide screen, identify CD19, 81, and 58 at the top intrinsic factor related to resistant to this drug in BLL. CD58 level in BLL clearly is very important for blinatumomab-induced T cell cytotoxicity against BLL. The PAX5 mutation in ALL directly leads to a complete loss of CD50 expression because of the inactivation of the PAX5-driven enhancer at the CD58 locus. And this eventually gives rise to leukemia resistance to blinatumomab. And also CD58 is essential for blinatumomab-induced T cell activation and proliferation. So I want to end by showing this slide because a lot of times people ask me, why do we still study LL pharmacology now with this disease curable for 95% of the children? And the answer, I tried to come up with a good answer, but I think now I have a very good response to that, because first time in a long time, we now have a wave of new and highly effective agents they're probably gonna really shift the paradigm of how LL is treated. For example, we have immunotherapeutics, including blinatumomab, inotuzumab, CAR-T, daratumumab. We have a number of tyrosine kinase inhibitors, ABL inhibitor, 3 inhibitor, LCK. We also have a number of BH3 mimetics that are very, very effective against the BLL and the TLL. So now we have really nice tools that we can use to think about how to develop the next generation of ALL therapy. And my hope, and I also hope I convinced you that pharmacogenomics is a very, very powerful and unique tool and weapon we have to help us guide the development of the next generation of ALL therapy here at St. Jude and elsewhere. So with that, I need to thank everybody working in my lab. I want to thank Mary Rowling, Bill Evans, and Jin Hong Pui, my 3 mentors here at St. Jude's. It's been absolutely wonderful working with them in the past 16 years. All the collaborators here at St. Jude, COG, MD Anderson, and also my funding sources. So I will stop here, Seth, and be happy to take a few questions if we still have time.
I think we do. Thank you, June. That was a fantastic lecture, and I'm sure we will see lots of questions coming in just a moment. The one that we have so far is about the dasatinib story relative to TALL, and specifically notes that dasatinib is a tyrosine kinase inhibitor that covers multiple tyrosine kinases, and specifically wonder whether there was any data on the impact of the Src kinase inhibition component of dasatinib relative to the efficacy in TALL.
Yeah, this is a question we also received from the reviewers when the manuscript was under review. The experiment we did was, so we worked with Kim Stagmeyer and Dana-Farber, look at the cancer dependency map, and we look at the TAL cell lines that are sensitive to dasatinib. We ask whether those cells are dependent on LCK versus other members of the Src family, and LCK is the only gene that's essential for survival in dasatinib sensitive TLL. We look at Src, we can look at LIN, we look at FYN, a bunch of other members of the family. None of them had any effects. We also did experiment where we were overexpressed a dominant negative LCK mutant version, and we showed that we can completely reverse the drug sensitivity phenotype. Again, pointed to the specific effects mediated by LCK activation.
Perfect, thank you. I see a few more coming in, but I'll take the advantage of being the moderator and ask a question of my own, which is, as you looked at the potential moderators of blinatumomab efficacy, I noticed that in regards to CD19, one of the molecules that appeared to have scored highly was IKZF1. And so wondered if you had thoughts on how that may have implications for the use of immunotherapy relative to pH positive or pH like ALLs, these groups that are frequently have mutations in IKZF1 and then also are historically very difficult to treat.
Yes, that's a very good point, Seth. I think as you and others know as well, IKZF1 genomic abnormalities are common in pediatric and also adult ALL, especially high-risk ALL. There are different types of IKZF1 lesion. They're the dominant negative fusion, and there are also point mutations. So what we don't know right now is whether mutation versus deletion have different impacts on CD19 expression. So I think that's something we need to do. We'll go back to the pan-ALL dataset to look at that more carefully. I suspect that they don't impact CD19 expression in the same way. And also there is the homozygous loss versus homozygous loss, whether that translates to differential level of resistance to blinatumomab.
Excellent, thank you. I think one question that sort of is touched on by one of our questions here is just the idea of whether the data that you're generating across subtypes gives us hints about either new classes of agents that should be brought forward or ways to use these new classes, the different classes of agents in different subsets of ALL. And I think you hinted that with your future directions of how we design new trials and how we design therapy for children. But thoughts on prioritization relative to the data that you are developing?
That's a big question. Probably requires another conversation. But I think there are two directions we can think of. One is for subtypes that are clearly resistant to cytotoxic drugs, say asparaginase or glucocorticoids. Do we want to continue to give those agents to patients? For example, ETP seems to be very, very sensitive to, very, very resistant to glucocorticoids. Do we want to give all ETPs glucocorticoids the same way that we give that drug to other TLLs? But if you look at ETP in terms of the glucocorticoid sensitivity, there is still some variation. So some patients, even with ETP, do show some level of response to glucocorticoids. So it will be a little bit difficult to justify the complete removal of glucocorticoid for ETP. But of course, the other direction, which I think is more exciting, is if a patient or a certain subtype is linked to sensitivity to new drugs, especially targeted drugs, do we want to give them that drug? And then if we do give that patient a targeted agent, do we use that to replace the existing cytotoxic drug, or do we give that on top of the cytotoxic drug-based treatment regimen? So at least for me, I would hope that we will be able to individualize therapy such that we can replace cytotoxic drugs. Some of them, for example, daunorubicin, which we know has long-term side effects. Replace those type of agents with more targeted drugs, either immunotherapeutic or tyrosine kinase inhibitors. But we will really need to have even bigger cohort than we have now to better understand the biological basis above and beyond just molecular subtypes before we can start implementing.
Perfect. Thank you. All right, we are at the top of the hour, and so I'll take this opportunity to thank you, June, for a fantastic talk, and thank everyone for joining us today. And this is a reminder that Dr. Yang has agreed to stay on for a little bit for additional questions afterwards, and so stay tuned for that. Before we jump into that extended Q&A, a quick reminder to join us next week on the 12th when Dr. Morton from the NCI will present on cancer susceptibility to subsequent neoplasm following childhood cancer. And with that little transition done, I will go back to the questions. There was a question about data for infant ALL and the data that you've generated in terms of pharmacogenomics for that difficult to treat population.
Yeah, I'm trying to remember now. We don't have a huge cohort of infant ALL cases in this data set. I think we probably have somewhere between 10 to 15. And they do seem to be more sensitive to FLT3 inhibitors, which has been reported previously. And will be we do plan to do a separate analysis specifically focusing on infant ALL, especially if we can get more cases for pharmacotyping.
Fantastic. Dr. Relling asks about the role of the niche in drug sensitivity and the ability to interpret these drugs with or without that interplay.
It's probably not from the assay that we have, because it's a co-culture system. I don't know how much difference well, first of all, I don't know how feasible it is for us to characterize the interaction between stroma and leukemia cell in the system that we're using now. We'll be able to, I think we'll have to characterize that on individual cases to ask the question whether that contributes to differences in drug sensitivity. I hope I understand Mary's question.
I think so. There's a question about TCF3 PBX1 and how Blina may play into frontline therapy. I think as a treating clinician, I would say that Luna is already entering frontline therapy. And so I think it will be interesting to see as data matures across the different groups that are using it, how it interacts with these individual genetic subtypes. Anything to add to that?
Maybe just one thing. I think the CD19 expression clearly is very, very important for blinatumomab response. And I think after once we have CD19 expression at a certain level, going higher doesn't necessarily make it better. So yes, TCF3 PBX1 has very high level of CD19, but maybe the response is just as good as some other subtypes with a little bit lower CD19 because it's not a linear correlation. That's just based on our data, based on our understanding.
A question about diagnostic and relapse pairs and the interaction between those and genomic subgroups.
Yeah, we actually just started analyzing that data, comparing diagnosis to relapse. Clearly, things like glucocorticoid is almost always more resistant, almost always less effective at relapse compared to diagnosis, probably because of acquired resistance to those drugs. For targeted drugs, we still need to complete the analysis to know whether the resistance to glucocorticoids is accompanied with the sensitivity to some other drugs. I think the question about hyperdeploying ETV6 from Trump, they are underrepresented in the relapse cohort. So I think we'll have to collect more samples to understand to have a better statistical power, so that we know whether there's anything specific to those subtypes at time of relapse.
The next question comes from Jatinder Lamba about the interaction between LC50 and either toxicity or efficacy.
We haven't analyzed the correlation with toxicity, but efficacy, at least for the cytotoxic drugs, is there's a very strong association with MRD. So I would say, yeah, the correlation is pretty good. But for the newer targeted drugs, because we haven't given them to patients, so we don't, it's hard to tell whether the ex vivo LC50 is correlated with their efficacy.
The most recent question is asking for your favorite publications to cite these differences across genetic subgroups.
I think I can, yeah, that. So it was asking whether there's a portal. Yes, there will be a portal. The plan is to get this data out there for everybody to mine and be able to correlate drug response or drug sensitivity with the genetic features. And it's just stay tuned, and we'll reach out to the scientific community when that happens, hopefully soon.
So the next question is, we use chemotherapy in combinations, largely to get around drug resistance, but also to mitigate toxicities. And so the question is it possible that combination therapies alter drug efficacy?
That's a very good point. I think it's definitely possible. The limitation, one of the significant limitation of our pharmacotyping assay is we don't have enough cells to test a combination. I think it will be really great if we can modify the assay that we have right now to enable two drug combination, for example, at least for some of them. That will be really nice, but we have to prioritize which drug pairs to be tested, and maybe even prioritize which subtypes we will use to interrogate that.
The next question asks about co-culture once again, and specifically asks about the role of cell cycle progression in drug response within the context of a co-culture assay.
We have not analyzed that data simply because we did not put a cell cycle marker in the imaging system. But I think that's something we can definitely do. I can imagine adding things like protium iodine to look at S phase, G1, G2, etc. And that might be interesting. I know that for drugs that are cell cycle dependent, especially thiopurine and methotrexate, the drug sensitivity is related to cell progression. But for targeted drugs, my guess is that they're less likely to be influenced by cell cycle. But it's a good idea to test that.
So I think the rate of new questions has slowed down a little bit. So I will say thank you once again, June. It's always fantastic hearing you talk and hearing the work you do. And today was clearly no exception. Very much appreciate you presenting, and look forward to seeing everyone next week.
Sure, thank you. There's one more question. You want me to answer that one, or?
Go right ahead. I don't know the answer to that one. So it's a
Okay.
question about whether there are companies designing therapies to target CD58 using CRISPR. My impression is that CRISPR-based therapeutics are still very early in their infancy in terms of pharmacological therapies. I don't have a better answer to that one than that. Don't know if you have anything else.
Nope, nothing to add. Well, thank you very much for having me. It was fun.
Thank you. Take care, everyone.